The resting membrane potential of most neurons is about minus 70 millivolts. Because the resting membrane potential is negatively charged, we say that neurons are polarized. This resting membrane potential is due to two things. Number one, the difference in ion concentrations inside versus outside the neuron. And a slide or two ago, I did mention that extracellularly, so outside the neuron, there is a higher concentration of sodium ions. And inside the neuron, there is a higher concentration of potassium ions. The second contributing factor to the resting membrane potential of neurons is the difference in permeability. So the cell membrane, we know, does not allow ions to flow directly across. Ions need to utilize ion channels. So the neuron can control the permeability of the cell membrane to these ions by controlling the permeability of these ion channels. We can look at changes in membrane potential as either making the membrane potential more positive or more negative. So if we look at this figure here on the bottom left-hand corner of slide 17, this blue line is representing the resting membrane potential of a neuron. And we see that it is resting at minus 70 millivolts. If we stimulate this neuron and we stimulate it with a depolarizing current pulse, the membrane potential of this neuron also depolarizes. So you see it's becoming less negative, and then it returns back to rest. So depolarization refers to a change in the membrane potential such that it becomes less negative or closer to being positive, right? Because it's going in this upward direction. A hyperpolarizing pulse or stimulus causes a hyperpolarization of the membrane potential of a neuron. So in this figure, again, the neuron is resting at minus 70 millivolts. The hyperpolarizing stimulus is applied, and the result is a hyperpolarization of the membrane potential. And hyperpolarization means more negative. Action potentials are how neurons conduct information over longer distances. Graded potentials are how neurons conduct information more locally. So if we look at this figure here on the bottom left-hand corner of slide 18, we are looking at a cross-section of a neuron. And you can see all these negative symbols on the inside of the cell, because remember, the inside of a neuron is negatively charged. It is resting at minus 70 millivolts. So the outside of the neuron is positively charged relative to that inside of the neuron. Here in red is an electrode that is delivering a stimulus. That stimulus causes this area of this cell to depolarize. So right here in this area, the cell shaded red, instead of being -70 like the rest of the cell, it is depolarized. So maybe it's -50 millivolts right in here. So graded potentials are potentials that spread locally. And we see that represented in this middle panel here. We can see this dark red center is where that initial depolarization occurred, again, from the figure on the left-hand side. But just as a drop or a spoonful of cream in your coffee will spread throughout the mug, this depolarization also spreads throughout this cell. We can see an example of the spread of this depolarization as related to distance in the graph on the right-hand side. So the most depolarization, or the largest change in membrane potential, happens right where the stimulus was applied. And as we move further away in either direction, the change in membrane potential is smaller and smaller, because the spread of this depolarization decays as it moves further and further away from the stimulus. Action potentials are for long distance communication. So neurons generate action potentials to send information a longer distance. So the action potentials are generated in neurons and in muscle cells. Those are the only two cells that we see action potentials. Action potentials look something like this figure on the bottom right-hand side of slide 19. So this horizontal black line here represents the resting membrane potential, which you can see is -70 millivolts. This vertical arrow here is representing a stimulus. So a stimulus is applied and it causes a short depolarization in the membrane potential, and then you can see a return to the resting membrane potential. So stimulus number 1 is the smallest. We increase the intensity of the stimulus for stimulus 2 and 3, and you can see the depolarization is a little bit larger the bigger the stimulus, until we reach some threshold, which you see here in red. So stimulus number 4 was strong enough for the cell to depolarize to the point of this threshold. Once this threshold is reached, an action potential is triggered, and the action potential you can see is this large upward and then downward deflection in the membrane potential. So action potentials consists of 4 stages: the resting stage, depolarization, repolarization, and hyperpolarization. And if we look at this figure here, initially we have the resting stage, then we have the depolarization, which is this whole upward deflection of the membrane potential, and you see it can go as high as +30, +40 millivolts. The repolarization is this downward deflection of the action potential as the cell goes back down towards its normal resting membrane potential of -70 millivolts. You see this downward deflection at the end of the action potential. This is called hyperpolarization, and then the cell returns to that resting membrane potential. So if we look at what is causing these different phases of the action potential, again, it goes back to those ion channels opening and closing. So at rest, the cell is at minus 70 millivolts. That is our resting membrane potential. At rest, all gated sodium and potassium channels are closed. The only channels that are open are the leakage channels. And remember, those leakage channels are what's helping maintain the resting membrane potential. In the second phase of the action potential, we see the depolarization. The cell membrane potential becomes less and less negative and actually even becomes slightly positive. This upward deflection of the membrane potential is caused by voltage-gated sodium channels opening. So on the previous slide, I mentioned this threshold for depolarization right here around minus 60 millivolts. So this threshold of minus 60 millivolts is important because these voltage-gated sodium channels will not open unless the cell reaches that threshold. So you can see the first three stimulations did not depolarize the cell to this threshold. So in these first three trials, no action potential was generated. In the fourth trial, the stimulation was strong enough for the cell membrane to depolarize enough to trigger the opening of those sodium channels. And remember, sodium is highly concentrated on the outside of a cell. So when a sodium channel opens, sodium will rush into the cell. Because sodium is positively charged, when sodium rushes in to the cell, it brings in that positive charge. More positive charges inside the cell are what are responsible for the cell becoming less and less negative and even reaching positive membrane potentials of plus 30 to plus 40 millivolts. The repolarization of the action potential is this downward deflection of the action potential. This is caused by inactivation of those sodium channels. So those sodium channels will inactivate or begin to close, and that means that sodium will no longer be rushing into the cell, which means we will reduce the amount of positive charge inside the cell. At the same time, potassium channels begin to open. Just like those sodium channels, these are voltage-gated channels. So they are triggered when the cell reaches a particular membrane potential. When the voltage-gated potassium channels open, potassium will leave the cell. Again, potassium is highly concentrated inside the cell. So when a channel that is permeable to potassium opens, potassium will leave the cell. So this means that there is fewer positive charges inside the cell, and that causes the cell membrane potential to become more and more negative. The last part of the action potential is this hyperpolarization part right here, and you can see it more clearly in this figure. So in hyperpolarization, the membrane potential becomes more negative than it usually is at rest, and then returns to the resting membrane potential. Some people call this the overshoot. The membrane potential is trying to return to rest, but it overshoots it just a little bit and then returns to baseline. And again, this change in membrane potential is due to the flow of ions. During the hyperpolarization, during this last part of the action potential, there are some potassium channels that are remaining open, and we see those voltage-gated sodium channels reset and become ready for the next action potential to occur. Action potentials can be measured by using electrodes, and there is an image of part of a neuron. I think this is supposed to be part of an axon, and there is an electrode placed into the axon. So this is one way to measure the membrane potential of a neuron. In this figure, I'm showing you an example of experiments that I used to perform back when I was doing research. I used a rat, an anesthetized rat, and lowered an electrode into the brain and then recorded neuronal activity from the anesthetized rat. This is what a recording looks like, and here you see in the small inset an action potential, and this line here represents 10 seconds of recording. And in this 10 seconds of recording, each vertical line is an action potential, as we've discussed. I'm going to play a recording of this experiment that I performed, and you will hear a series of pops. And each popping sound is an action potential. And I'm showing this to show you that this process happens very quickly. You can see an action potential takes around 2 milliseconds to happen, or even a little bit less. So let's listen to this. Oops. Let's listen to this. Okay, so those popping sounds were action potentials. So we heard a whole bunch of action potentials being generated by a neuron. In this video, we are looking at a recording performed by O'Keefe et al. And here is an image of a maze, and right here in the green circle is a rat. So as the video plays, you will see the rat walk along the maze and follow the path. And as it walks through this maze, you will hear as well as see action potentials that have been recorded from the hippocampus. And these are color coded, so this is showing you what will show by the end of the experiment. And these different colors are representing different neurons. So as the rat moves through the maze, I encourage you to look at when each different neuron is firing. Okay, so just like the previous slide, those popping sounds were action potentials, and they are color coded. So you see in this part of the maze, this dark blue neuron was active. The red neuron seems to be active here and maybe a little bit up here. And again, these are recordings from the hippocampus. These cells are called place cells. Place cells convey information about location. We have place cells in our hippocampus as well, and a similar experiment has been done using virtual reality taken from individuals who had electrodes implanted to alleviate neurological conditions like seizures. And place cells relay information about location as well as movement. So for example, in this figure, we see this dark blue neuron appears to be active as the rat makes a right-hand turn. And up here, we see dark blue again because the rat actually turned to the right before continuing on in the maze. So these aren't hard and fast rules, but they do have information with respect to place. In this experiment, the Higgins lab is recording from a dragonfly. So in the video that plays, you will see a dragonfly that is in front of a computer monitor. And the computer monitor will show a visual stimulus. You'll see a black square travel along the computer screen. And we are looking at the recordings of the dragonfly responding to that visual stimulus. Okay, so a little bit more about action potentials. As I mentioned, not all depolarizations will trigger an action potential. That depolarization must reach the threshold in order to trigger an action potential. Action potentials are all or none. An action potential either happens or it doesn't, which is why I have Mr. Yoda right here. You might be familiar with Yoda. He says, do or do not, there is no try. So a neuron will fire an action potential or it won't. There is no try. Action potential propagation refers to the action potential being transmitted down the axon. Remember, there are some neurons whose axons are a meter or even longer. So this propagation is traveling, in some cases, very long distances. Action potentials are propagated in myelinated axons through saltatory conduction. So if we look at this top left-hand figure on slide 26, we are looking at an axon. And we are seeing a stimulus being applied here at this red line, and that's inducing some change in the membrane potential. This is representing a non-myelinated or unmyelinated axon. And what we see is that graded potential. Again, the change in membrane potential is graded. It decays over some distance. We saw this several slides ago. So every so often, the axon has more of those voltage-gated ion channels to restart that change in membrane potential. So you can see this helps the signal propagate or travel down the axon. Well, in myelinated axons, here we see myelin here in blue, myelin actually insulates the axon, which means the membrane potential does not decay as quickly. So you can see this top figure, it decays very quickly, but this myelinated axon, the change in membrane potential does not decay nearly as quickly. There are gaps in the myelin sheath, and that is where we see more of those voltage-gated ion channels to restart the action potential. So the action potential appears to jump from gap to gap to gap down the myelinated axon. So these gaps in the myelination are called nodes of Ranvier, and the conduction of an action potential jumping from one node to the next node to the next node is called saltatory conduction. The advantage to saltatory conduction is, number one, it's faster, and number two, with fewer of those voltage-gated sodium channels, we have less sodium entering the axon in order to convey the same information. And this is important because the cell has to spend energy to maintain the appropriate concentration of sodium inside the cell. So after an action potential occurs, the cell has to use energy to pump those sodium ions back out of the axon. So by using saltatory conduction, we allow fewer of those sodium ions into the cell, and thus means we have less sodium ions we have to move back outside the cell once the action potential is done. There are two things that affect the rate of propagation of the action potential down the axon. Number one is the diameter of the axon. Larger diameter axons have less resistance. So you can imagine if you had two different garden hoses, one had a 2-inch diameter and another had a 1-inch diameter, the water would flow more easily through the garden hose with the larger diameter. The second factor that affects the conduction velocity, or how quickly an action potential is propagated down an axon, is the amount of myelination present on the axon. So if there is no myelination, the axon is relying on graded potentials. However, if there is myelination present, the axon can utilize saltatory conduction.